Okay, so welcome to this next video in which we are discussing insulin synthesis and secretion. Okay, right. What we now want to discuss is how a change in electrical potential difference, namely a depolarization, which is what we've seen has occurred so far due to the ATP closing the K-ATP channels, is going to lead to a calcium signal within the cytoplasm of the beta cell. Okay, and it's going to do this by activating voltage-gated calcium channels. So let's now discuss the structure of voltage-gated calcium channels. And for short, voltage-gated calcium channels are abbreviated to VGCC. So the V is for voltage, the G is for gated, and then the CC is for calcium and then channels. Okay, so voltage-gated calcium channels. Now basically, voltage-gated calcium channels consist of a main subunit, which is known as the alpha subunit. So let me show this first. Okay, so we'll have the main subunit sitting here. And this is the subunit that actually has the pore within it that allows the calcium ions to actually move across the membrane. So here is the main subunit here, okay, and this is what's known as the alpha subunit, okay, and then you have a bunch of other subunits attached onto it known as the accessory subunits. So for instance, you'll have a beta subunit which is attached down here, okay, on the cytoplasmic side of the alpha subunit. You'll have a gamma subunit that sits within the membrane alongside the alpha subunit. And then you'll have an alpha-2 delta subunit here, which consists of the delta subunit, which is the portion which spans the membrane, and the alpha-2 portion up here. And the reason they're not considered two separate subunits is because they come from a single protein that is then cleaved apart, very much so like we saw pre-pro-insulin makes both the A chain and the B chain of insulin. Basically, the alpha-2 and the delta portion of the alpha-2 delta subunit come from the same protein that is cleaved up and then attached back together. Okay, right. So, by the way, this subunit down here, which was attached to the cytoplasmic side of the alpha subunit, is known as the beta subunit. Okay, so the type of voltage-gated calcium channel that you have within the membrane of beta cells is known as an L-type voltage-gated calcium channel. And I want to explain to you what it means to be an L-type voltage-gated calcium channel. Basically, when we call a voltage-gated calcium channel an L-type voltage-gated calcium channel, it doesn't refer to the alpha-2 delta subunit, it doesn't refer to the gamma subunit, it doesn't refer to which beta subunit you have. It refers completely to the type of alpha subunit that you have, the most important subunit of the voltage-gated calcium channel here. Okay, so basically there is not just one alpha subunit. There are in fact 10 different alpha subunits, and these are split into 3 separate families. So there is the CAV, which stands for voltage gated, that's the V, and then calcium channel, and then there's the CAV1 family, which has 4 members within it, which are the CAV 1.1, CAV 1.2, CAV 1.3, and again it's got a beautiful naming system, and then finally CAV 1.4. Okay, then we have the CAV2 family, which has 3 members within it, the CAV 2.1, CAV 2.2, the CAV 2.3 family, CAV 2.3 protein, and then finally we have the CAV3 family, which again has 3 members. So it has the CAV 3.1, the CAV 3.2, and the CAV 3.3 proteins. Okay, so these 10 different proteins are all alpha subunits of voltage-gated calcium channels. Okay, now if you use one of these protein subunits to make your alpha subunit of your voltage-gated calcium channel, then you are called an L-type voltage-gated calcium channel. So if you use a member of the first family of CAV proteins, okay, so if you use a member of the first family of alpha subunits, then you're called an L-type voltage-gated calcium channel. Okay, right, and just to conclude our discussion of voltage-gated calcium channels, I just want to show you the membrane-spanning topology of an alpha subunit of a voltage-gated calcium channel. Now these things are quite massive. If you thought the sulfonylurea receptor was a nightmare, look at this. Okay, so basically it's going to have 4 separate domains, and all of these domains are effectively repeats of one another. So, wait, have I done it wrong? What have I done wrong here? I'm drawing a G protein coupled receptor. Yes, this is wrong. We're going to have to... There's no way I can rescue that. No. Scrap this, okay, and start again. I needed a bigger membrane anyway. It won't fit on there. Okay, so here is the lipid bi-there. The amino terminus should be on the cytoplasmic side. So here is the amino terminus. Okay, I suppose I could have flipped it up over, but that would have been horrendously confusing. So it's better I just start again. Then we have 4 membrane-spanning alpha helices, a 5th one, then a p-loop, then the 6th membrane-spanning alpha helix. So that's domain 1 complete. And then you repeat it all over again. So 5 membrane-spanning alpha helices, a p-loop, that's domain 2 complete. Then we have domain 3. Okay, 5 membrane-spanning alpha helices, a p-loop, a final membrane-spanning alpha helix. And then we have domain 4. Okay, so basically... that now concludes our structure of the alpha subunits. You have domain 1 here, domain 2 here, domain 3 here, and domain 4 here, and they're basically repeats of one another like so. Okay, and these will fold up to make an alpha subunit, and there are 10 different types, and they're all, every single one of these 10 different types, they all look like this, and they all fold up to make a pore, basically, through which calcium can move. And then the different subunits attach on. So, for instance, the beta subunit attaches to this cytoplasmic loop between the first and second domain, so the beta subunit will attach on here. Okay, right. Now the important thing is that these channels are activated by a depolarization in the electrical potential difference across the membrane. So when the electrical potential difference across the membrane is depolarized, they will be activated to open, and then when they open, calcium will move from the outside into the inside of the cell, and the reason is quite simple, basically. The concentration of calcium extracellularly is around 1.5 millimolar, okay, which looks pathetic compared to, for instance, the sodium concentration extracellularly. But then let's look at the concentration of calcium intracellularly. It's around 100 nanomolar. That means that the concentration extracellularly is 15,000 times bigger than the concentration cytoplasmically. Okay, so there is a 15,000-fold concentration gradient across the cell membrane for calcium. That dwarfs the concentration gradient for sodium, which is around 12-fold, and even the concentration gradient for potassium, which is around 40-fold. Okay, so as soon as you open that voltage-gated calcium channel, calcium is going to move in from the extracellular fluid into the cytoplasm, and this calcium is then going to help cause the release of the secretory vesicles, which are full of insulin and C-peptide. Okay, but you also need something else which is also activated by ATP. Okay, and basically this is the production of certain arachidonic acid metabolites. So let me get a new piece of paper, and then let's talk about these arachidonic acid metabolites that are important in releasing insulin and C-peptide secretory vesicles. Okay, so basically, when the ATP goes up in the cytoplasm of the cell, it is believed that it activates something that is not well understood yet, but which has a name regardless of the fact that we don't understand it very well. Okay, and this is known as the ASCII, ASCI, phospholipase A2, so PLA2. So this is the symbol name for it, but what does this actually stand for? So ASCI stands for ATP sensitive, that's the AS, so A is for ATP, and then the S is for sensitive, and then the CI is for calcium insensitive. Okay, so this is the calcium insensitive, because usually phospholipase A2 enzymes are calcium sensitive. Okay, so usually they have C2 domains, and when the calcium binds to their C2 domain, that activates the translocation of the phospholipase A2 enzyme from the cytoplasm to the inner leaflet of the phospholipid bilayer, where it will then act on its substrate, which is in the phospholipid bilayer. But this form of phospholipase A2 is going to be insensitive to calcium, and instead is going to be activated by ATP. Okay, and then PLA2 is just short for phospholipase A2. So PL is phospholipase, and then it's phospholipase A2. So, what's going to happen is when ATP goes up, this ATP-sensitive, calcium-insensitive phospholipase A2 is going to be activated, and this is going to lead it to act on a substrate which is within the phospholipid bilayer of the beta cell of the islets of Langerhans. So let's discuss its substrate. Now, its substrate is something known as phosphatidylcholine. Okay, and in order to understand what phosphatidylcholine is, we need to understand what the structure of a phosphatidate molecule is. So basically, phosphatidate is another word for a phospholipid. It's the old name for a normal old boring old phospholipid. So what I want to convince you of basically is that phosphatidylcholine is just a modified phospholipid. It's just a phospholipid with a normal or boring old phospholipid with a choline molecule stuck onto it. So let's start off by revising the structure of a normal old boring old phospholipid. Okay, so in cartoon form, we'll show the structure of a normal old boring old phospholipid like so. Okay, so these vertical lines here, these represent the long-chain carboxylic acids, or the fatty acids, which are esterified to the first and second alcohol groups of the glycerol molecule. Okay, so these are fatty acids, which is the biochemist's name for what a chemist would call long-chain carboxylic acids. Okay, so both names are valid. Okay, and these are just carboxylic acids which have incredibly long hydrophobic tails, which don't interact well with water and therefore interact well with one another, and that makes up the hydrophobic core of the phospholipid bilayer. Okay, now these two long-chain carboxylic acids, they are esterified to the first and second alcohol groups of the glycerol molecule, which is this horizontal line in green here. So this is glycerol. Okay, and the proper chemist's name for glycerol is to call it propane 1,2,3 trial. And although propane 1,2,3 trial is a mouthful compared to glycerol, it's a useful name because it tells you exactly what we are dealing with here. It tells us that we're dealing with a free carbon hydrocarbon molecule with alcohol groups coming off each one of the carbons of that molecule. So you have 3 alcohol groups, one off each of the carbons. Now, attached to the first and the second alcohol groups of the glycerol molecule, you have these long-chain carboxylic acids coming off, but attached to the third alcohol group of the glycerol molecule, instead you have a phosphate group, and this is the phospho in the name phospholipid. Okay, and this phosphate group is attached to the alcohol group of the third carbon of the glycerol molecule via a phosphoester link. Now let me just show you the structure of a phosphate group. So a phosphate group has a phosphorus atom at the center, and this phosphorus atom has an oxygen double bound to it, and then it has 2 alcohol groups coming off it, like so, and then it has a single bond to an oxygen which has acquired another electron by ionic means and therefore has a negative charge. Okay, so this is the structure of a phosphate group. And what I would encourage you to think of this as being analogous to is a carboxylic acid group. Because if we replace this phosphorus atom with a carbon atom, ignore the fact that it would make no sense because you'd have 5 bonds. But if we did replace this with a carbon and just looked at this portion here, that would be a carboxylic acid group. And effectively you can link this phosphate group to an alcohol group in exactly the same way as you can link a carboxylic acid group to an alcohol group. Now it's no longer called an ester link, instead it's called a phosphoester link. And what's incredible about this is even once this alcohol group is involved in a phosphoester link to the alcohol group of the glycerol molecule, you'll still have an alcohol group over here which will have effectively a carboxylic acid group, but with a phosphorus atom instead. And that can then link to another alcohol group, and that's what's going to happen. So this is the structure of a normal phospholipid, or a phosphatidate molecule. Now, in order to make phosphatidylcholine, what we are going to do is add on a choline molecule which has an alcohol group onto the free alcohol group of the phosphate group here. So we're going to stick a choline molecule onto this phosphate group here. Okay, and I'll just draw this as a blue box. I'll show you the structure of choline in a moment because it's the same choline as is present in acetylcholine. I think I'll just fill in that box there. Right, so this is choline here, and the whole structure then is now called phosphatidylcholine. Phosphatidyl because we've got a phosphatidate molecule attached to choline. Okay, so let me show you the structure of choline then. So basically, choline is a 2-carbon molecule with an alcohol group of it. Okay, and also an amino group which has 3 methyl groups coming off it. So over here you have a nitrogen atom which then has 3 methyl groups coming off it, like so. And of course then the nitrogen has 4 bonds coming off it. It should only have 3. So one of these bonds consists of the nitrogen atom donating both electrons to the other member of the bond. Okay, so for instance, let's say it's this one here. This covalent bond will have 2 electrons in, both from the nitrogen. Now, the understanding of a covalent bond is that 1 electron has come from each of the members of it. So when the nitrogen donates both electrons, it's as though it has donated 1 to the carbon, and then the carbon has given that electron back, and the nitrogen has provided a second electron to form a covalent bond. So basically, it's a kind of horrible trick the carbon has played on the nitrogen. It's sort of taken one of the electrons from the nitrogen and then has sort of said, okay, let's make a covalent bond with this electron that I've just nicked off you. Okay, so that means that the nitrogen effectively gets a positive charge. So you might then ask, well, will the carbon then have a negative charge? And the answer is no. The carbon that originally came along will have had a positive charge to begin with. So when it nicked the electron off the nitrogen, it neutralized itself and passed the positive charge effectively on to the nitrogen. So this then is the structure of the choline molecule. And it's this alcohol group of the choline molecule that will be attached to the free alcohol group of the phosphate group that is on the phospholipid molecule. Okay, now hopefully what you can appreciate is that this phosphatidylcholine molecule, effectively, it's just a phospholipid with a bigger head. We've just stuck an extra group onto the head of the phospholipid. Okay, so it's just a modified phospholipid. Now, this phosphatidylcholine molecule is the target for the enzyme ATP-sensitive calcium-insensitive phospholipase A2. In fact, it's the target for all phospholipase A2 enzymes. And what basically this ATP-sensitive calcium-insensitive phospholipase A2 will do is it will break the bond between the second carboxylic acid molecule and the glycerol molecule to yield 2 products, both of which will remain in the phospholipid bilayer. Okay, so one of the products is going to be a phosphatidylcholine molecule, which is here. Oh, sorry, not a phosphatidylcholine. What's left of the phosphatidylcholine? Phosphatidylcholine was when it had both long-chain carboxylic acids. It's not called phosphatidylcholine anymore. This now is called lysophosphatidylcholine. Okay, so we've removed one of those carboxylic acids, the second one, which is why it's phospholipase A2. The 2 tells you that it's the second carboxylic acid that you'll be removing. So this now is called lyso, and it should be one word, so I'll just put PC for phosphatidylcholine. People often do that. I was just foolish, and I'll write it now down below. I didn't leave enough space to put phosphatidylcholine because it's all one word, so I would like to write it as such. So this is a lysophosphatidylcholine, And then you also have within the phospholipid bilayer the free long-chain carboxylic acid that was originally bound to the second alcohol group of the glycerol molecule. Okay, now the general long-chain carboxylic acid that you have bound in the second position of a phosphatidylcholine molecule is arachidonic acid. Okay, and it is this liberated arachidonic acid that is now going to be used to produce certain arachidonic acid metabolites that will promote the release of insulin. Okay, and we'll continue this discussion in the next video.